Good day, everyone. I'd like to welcome you to class this day. We'll be looking at CSP 530, and I still remain your friend, Emeka Chidebe, and we'll be taking on this course this day. I'll be looking at plant disease, also known as phytopathology. Now, in the course of our class, we will look at these following learning outcomes. Some of the learning outcomes we'll be looking at is 1, to understand the basic terminologies associated with plant pathology. We would like to identify the common diseases affecting crops in the tropics, and then we study the principles and methods of disease control. In the course of this class, you will want to know what is plant pathology. And on a simple note, plant pathology, in some texts, you will see them written as phytopathology. It is a Greek word that means phyto stands for plant, pathos means suffering or diseased, while logos stands for study. So, putting it together, you can assume that it is the study of suffering plants or the study of diseased plants. Technically defined, it is a branch of agriculture, botany, or biological sciences which deals with the study of the cause of diseases, the resulting losses, and the control of plant disease. When you look at your screen, we have a figure of a tomato that has some disease parts, and below the figure you also see the micrograph of this disease after it has been cultured. So these are some of the things we'll be learning in the course of this class. Now we also want to know, how do pathogens affect plants? Now there are many ways in which plant disease affects plants, and there are many ways this occur. One is by utilizing the host cell content, meaning they dominate the cell content of their hosts. Secondly, by killing the host. Most of the times this is what is happening, and other times they interfere with the metabolic processes through their enzymes and toxins. Then thirdly, they weaken the host due to continuous loss of nutrients, or they interfere with the translocation of the food, mineral, and water. When we look at the figure by our left, you will see a tomato plant standing, and you can see the corner you have your place where there are diseased leaves. So these are how these plants, and they happen by what? By suppressing the chlorophyll content, and they can reduce the leaf area. You can see the area we shaded, and they can curb the movement of solute and water through the stems. They sometimes reduce the water absorbing capacity of these roots. They suppress the translocation of the photosynthesis away from the earth, and they sometimes promote wasteful use of products of photosynthesis, as in the formation of gauze, especially in your xylem and in your phloem structure of it. Now let's look at the scope and importance. Why is plant disease important? Plant pathology deals with different aspects of plant diseases and has a wider scope than the human pathology, which only deals with one aspect. In plant pathology, we have aspects such as plant virology, plant bacteriology, plant mycology. We have nematology. So these are the different, we'll call it aspects of phytopathology. In recent years, pathologists have began to specialize in particular aspects. The field in which notable advances have been made are the interaction between the host and the pathogen, then at chemical, molecular, and genetic level. Then we look at the plant virology, the chemistry of fungi toxicity, and we'll also look at disease forecasting. These are the areas notable advances in plant disease. Now on the figure we have here, you can see a grape. The disease part has changed color from the normal green to a lighter yellow. While if you look at the figure behind, you can see some root galls below the soil surface, while the fruits have lost their portion. They are now having mass of spores instead of having fruit in them. Then if you look at the trunk of the tree, you can see there are depressions where the stem has been deformed or distorted, as the case may be. Now going forward, how do we look at plant pathology today and tomorrow? Is a very wide scope, but advances have been made in the diagnostic and detection techniques, helping in accurate and quick detection and identification of these pathogens. Now, there are so many technology that has played in recent times in the detection of these diseases, some of which are ELISA, all these are acronyms. ELISA, monoclonal antibodies, the DNA probes, the PCR-based, and so on and so forth. Now, if you look at your figures, you will see we try to get some pictures of a PCR, what a PCR looks like. That is a polymerase chain reaction, and we also try to bring out the DNA probes. So in management of these diseases, there are also advances like in gene sequencing and gene silencing, the SAR, and other things. Now look at this tetrahydral. You can see we have here where the pathogen, the host, the environment, and the time at which it occurs plays a critical part in disease development. We call this disease triangle. In other way, some call it disease tetrahydral. Now, this is not where it ends. I encourage you to look up further. We sourced this material for further references. You can read up from Plant Pathology, the 5th edition by George A. Gross, and you will enjoy it when we meet next time to discuss further. Thank you, and do have a blessed day.